down to my fish room, I get a clutch of eggs. It actually doesn't hurt the mother, and then I artificially inseminate them, and I inject this DNA directly into the cell as it's starting to develop, the very first cell of the fish embryo. And then so the way this works is, if my fish cells have this plasmid in them, if they're using that enhancer to express the associated gene in that cell, it will glow green. Does that make sense? Yeah. So then I'm able to look at basically my fish and see where this enhancer is being used and what tissues is regulating this gene. So my work this summer was for a very specific gene. I was identifying these enhancers and looking at their expression patterns. And they're super cool. So this is one of the enhancers I looked at, and it shows in the fish that I was injecting that this midbrain comes on every single time. And what that means is that this enhancer for this specific gene is turning on expression of that gene in our midbrain. But these are literally fish that are just glowing, which I think is really, really cool too. And I also saw a different enhancer that was turning on expression of this specific gene in pectoral fins in my fish. And that's kind of cool because this gene I was looking at, we already know is involved in appendage growth for fish, so that kind of makes sense. And then also in the midline fin mesenchyme, which basically just means my tail fin tissue, which also makes sense for appendage growth. So it's really, really cool. I was able to identify some of these regions that are involved in regulation, and I did see them working in this way that I thought they might, where one enhancer is causing expression of that gene in one specific tissue type. Yeah.
So each enhancer is connected to a specific part of the fish, right?
That's what I was seeing.
So
yeah.
like when you put it in the injection or the midbrain, you know what specific enhancer it was connected to?
I did not explain that very well. So basically, I took an enhancer and I put it into the fish and I saw that glowed green in the midbrain, right? So what that tells me is that it's turning on expression of that gene specifically in the midbrain. Right. Does that make sense? Right. Okay. This is also like a very, very tough concept. I did not learn this until like a year or two ago. But basically, what I do is I learn about how genes are regulated, and I do that through looking at how these expression patterns work. And the thing that I think is really, really cool about it is that I actually have to make DNA, and I have to insert that DNA, and I have to look at how that DNA is expressed. So I work with something called transgenic DNA, where I'm actually modifying the DNA of the organism. And that brings me to CRISPR, which is really cool. So the current work that I'm doing is with CRISPR. This work is not with CRISPR because I was like randomly inserting this DNA I was making into the genome. I was just kind of throwing it in there. But do you guys know a little bit about CRISPR? A little bit.
Give the background.
I will give the background for sure. We'll keep it real simple. So CRISPR, basically what it does is it allows me to stick a piece of DNA into a very specific piece of the genome. So I can pick a place that I want to stick a piece of DNA in the genome, and I can stick that DNA directly into that place. Does that make sense? The other thing it can be used for is you can remove a piece of the genome anywhere you want to. And that's much easier than putting in a completely new piece of DNA. And it's a really, really powerful technology because it allows us to modify genes. We can knock out genes completely. Like if you have a gene that's causing a disease, you could literally go in there with CRISPR and just erase that gene. And then what I do is I go in and I can put genes in as well. And when I go, it's because my lab actually didn't tell me that in the model organism I was working on that had never been done before. So that's basically what my project has been, is I've been doing some of the first CRISPR, what are called knock-ins, where we're throwing DNA into the genome in a specific place that have been done in sickle back before. Tracking? Mm-hmm. Yeah? Okay, cool, cool. Yeah, please do stop me if I'm going too fast or if there's something that isn't. But this is probably the thing that I'm most excited about is CRISPR, because there's an amazing amount of potential for it, not only for disease, but a lot of the work I want to do is related to creating more diversity in already existing genomes of species using this tool, actually going in and causing mutations at specific sites. And I'm really interested in coral, which is why the marine biology stuff was up earlier. And we know that coral bleaching is happening really quickly as the climate's warming, but we also are identifying genes that are involved in that, and we're able to modify those genes now to see if we can actually prevent that. Yeah.
When you're like using CRISPR,
Yeah.
is it something where like, let's say a human has cancer, I don't know what the like CRISPR machine, is it like a machine or is it like a?
Yeah. So it's literally a piece of RNA.
Oh, okay.
It's really weird. So it's all at the molecular level, and basically what we're doing is we're putting a cocktail of the right types of proteins and RNA together, and because of the structure of these proteins, it's able to use a piece of RNA to find a different place in the genome by matching up to it correctly. It makes a little break, and then it will either get rid of the gene that way, or we can stick a piece of DNA into that break.
Oh, okay.
Does that make sense?
Like, you don't need to physically like take a cell and like do something with it and then put it back. You can just do it on anything.
It gets really complicated. You can do that. So people are starting to use CRISPR in therapeutics in that way, where they'll actually remove cells from a person and they'll modify each individual cell with CRISPR and then stick those cells back into the person, which I think is what you were asking about. But what I do is I just take an individual fish, and in the very first cell that's developing, I insert CRISPR and it will know where to go into the nucleus, and it will make that cut and stick in the DNA that I put with it. Yeah.
So is CRISPR relatively new, or is it?
That's an interesting question. Yeah. It being used the way that it's being used is very new. The actual new part about it is not the system itself. CRISPR actually, we didn't develop CRISPR. CRISPR is naturally occurring in bacteria, which I know sounds kind of weird, but it's actually an immune defense mechanism in bacteria that we recognized and started researching and realized that it could be co-opted for this purpose. And so that usage of it is brand new. But I would say it's accelerated dramatically in the last 5 years the ways in which people are using it, because it is able to knock out genes and knock in pieces of DNA. It's also able to, if I say epigenetics, how many of you have heard this term before? Okay, so there's another layer of genetic regulation on top of how genes are turned on and off that we can modify with CRISPR too. So we can actually like modify the regulatory machinery of genes as well. Yeah.
Does CRISPR stand for anything, or is that just a term?
No,